In this video, we will discuss about role of Biglycan in immunomodulatory. So before we go into the topic, let us understand this Biglycan in brief. Biglycan is uh, uh, the protein is called Biglycan. They have two chains of glycoaminos, glycan. So this Biglycan can be act to trigger this immunomodulatory. That three different categories are being shown here. So first thing, they have shown that the Biglycan interacts with this TLR2 and TLR4, and then in the macrophages, then it can lead to the activations of cytokines such as TNF-α, CCL2, and they will help in the recruitment of neutrophil through this interleukin-1 beta. And one more thing can occur is that if they interact with this P2X, then it will also leads to the NRLP3 and caspase 1 intercellular triggering that will lead to the formation of interleukin-1 beta, and it will also helps in this neutrophil recruitment. And if there is soluble Biglycan, then it can interact with this TLR2 and TLR4 in such a way that it will lead to the activations of cytokines such as IFN-β. This IFN-β helps in this T-lymphocyte recruitment. So this way, this Biglycan can recruit neutrophils and also T lymphocytes through the interactions with TLR2, TLR4, P2X, and in the macrophages. The other things is that Biglycan also determines the complex formations of CD14, TLR2, and TLR4. Based on the complex formation, it will trigger the downstream signaling such as this HSP70, TNF-α, CCL5, and they will ultimately leads to either activations or other recruitment of the other immune cells or activations of the dendritic cells. And the third category is about the Biglycan interacts with this TLR2 and TLR4. Not only just it activates the cytokines formation, but it will also activate the NADPHase, some of the NADPHase enzyme such as NOX1, 2, 4, and this NADPHase enzymes activations can either helps in this interleukin-1 beta formation or prevent the interleukin-1 beta formation, depending on the type of interactions that it has. And at the same time, what will happen that this NOX1, 2 also helps in the stimulation of, also helps the dendritic cells and macrophages towards this cytokine formations, this CXCL13. So these are some of the common roles of the Biglycans in this immunomodulatory, and this plays an important role, especially in this tumor microenvironment where macrophages are usually diverted towards M2 type macrophages, where it, instead of acting as a tumor suppression, those M2 type macrophages, macrophages which is converted to the M2 type, act as a tumor proliferations by suppressing the activated T cells that we have discussed in detail in one of the examples where we talked about Glycan interactions with its ligand on the pancreatic cancer cells. So this is just a big overview picture that what are the cytokines that Biglycan can triggers and on when they interact with these immune cells. So here the positive things have been shown here, that it will recruit the other immune cells such as neutrophil and T lymphocyte, whereas in the case of tumors or in the case of tumor microenvironment, those interactions might suppress the immune recruitment. At the same time, it will inhibit the activations of the T cells, thereby the T cells cytotoxic killing by CD8 plus T cells will be suppressed, as the lytic granules that are released by CD8 plus T cells will also goes down because of the deactivations or inhibitions of the further activations of the T cells. So I hope you understood in brief the role of Biglycans in the immunomodulatory if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.